morning and good evening to our colleagues and partners. Welcome to the first of the ASEAN Dx Preparedness webinar series. My name is Sidney, CEO of DXD Hub and co-chair of the ASEAN Dx Initiative. First and foremost, I would like to thank the panelists for taking the time to share their experience on this ASEAN Dx platform. I also would like to thank all of you for participating. We have over 450 registrations to this webinar, so we look forward to having a very exciting discussion on the topic of alternative testing platforms. So basically, we hope that through this preparedness webinar series, we can seed ideas and stimulate conversations amongst labs and practitioners to think about innovative solutions and alternatives so that we can better deal with this COVID-19 pandemic, which is far from over, unfortunately, and also to prepare for what may come next. So before we move on, just some administrative details. The webinar is recorded and the link to the recording will be sent to your email. There'll be a poll at the end of the webinar. It's just to help guide us in preparing for the next topic of your interest in this webinar series. So result of the poll will be made available on the ASEAN Dx website. So it is now my pleasure to invite co-chair of ASEAN Dx Initiative, Dr. Jaime Montoya, to give the opening address. Dr. Montoya is the Executive Director of the Philippines Council for Health Research and Development, Department of Science and Technology. Dr. Montoya, please.
To my ASEAN Diagnostics Initiative co-chair, Dr. Sidney, to our panel chair, Dr. Benedict, resource persons, Dr. Kristine Alvarado-Dela Cruz, Dr. Eric Vail, Dr. Wong Dr. Joseph Yao, guests from our ASEAN member states, and everyone joining us virtually today. Good morning. It is my honor to welcome you all to the first installment of the ASEAN Diagnostic Preparedness webinar series. COVID-19 has undeniably reiterated the importance of diagnostics in managing major health crises. It has shown us that the first step in addressing any disease is early and proper diagnosis. Endorsed through the 76th ASEAN Committee on Science, Technology, and Innovation in 2018, the ASEAN Diagnostic Initiative is a body that focuses on preparing diagnostic solutions in Southeast Asia for better healthcare in the region. Leading this initiative, the ASEAN Network for Drugs, Diagnostics, Vaccines, and Traditional Medicines Innovation, or the ASEAN NDI, and the Diagnostics Development Hub launched this webinar series in response to the increased demand for diagnostic technology and innovation during the pandemic. Joined by our experts from the Philippines, Singapore, and the United States, we hope that this activity becomes a platform for knowledge sharing and meaningful discussions. By sharing our practices, our best practices, insights, and ideas, we are able to build on each other's experiences to forward advancements in health diagnostics, to support and strengthen health systems and health resiliency in ASEAN member states. Before we officially start this webinar series, allow me to highlight why efforts such as this is key to managing this pandemic. Borrowing words from the World Health Organization, none of us will be safe until everyone is safe. Indeed, we must work hand in hand and empower one another to make sure that everyone is protected from COVID-19. This, I believe, is one of our strengths in the ASEAN Diagnostic Initiative, our solidarity. With this, in behalf of the ASEAN Diagnostics Initiative, I would like to thank you all for being here today. Your support is crucial, not only in forwarding diagnostics in the ASEAN region, but also in strengthening our health systems long term. We will continue striving to create platforms that will produce greater health outcomes for all. May we all have a productive session ahead, and we hope to continue seeing you all in the following and future webinars. Thank you, and may we all stay in the best of health.
Thank you, Dr. Montoya. Without further ado, I'll hand it over to the chair and moderator of today's webinar, Dr. Benedict Yen, Director of the Molecular Diagnosis Center at the National University Hospital of Singapore. Ben, please.
Thank you, Dr. Sidney. So thanks, Sidney, for the introduction. So I have the pleasure of chairing and moderating the session today. So I will now start the presentation session. In the interest of time, my introductions will be short, right? But I just provide the broad overview of what Sidney, myself, the DXD team have conceptualized in terms of the speaker presentation. So we'll start off by having sort of a more broad overview. So we have Dr. Joseph Yao and Dr. Kristine presenting about the testing landscape in their own regions. And subsequently, we'll go down to sort of more technical or laboratory-related presentations by Dr. Eric Vail and Dr. Wong. So I will start off by introducing our first speaker, Dr. Joseph Yao. He's the chair of Mayo's Clinic Division of Clinical Microbiology and Associate Professor of Laboratory Medicine and Pathology. He will provide an overview of COVID-19 and SARS-CoV-2 diagnostic landscape and testing methodologies in the US and especially in his center, Mayo, and explain what direct PCR is. Over to you, Dr. Joseph.
Thank you, Dr. Yan, for the kind introduction. In the interest of time, I'm just going to jump right into the presentation. Okay, I think you're seeing my slide okay? Good, all right, thank you. First, let's take a look at the current definition of COVID-19. Back in March 2020, WHO defined a case of COVID-19 as a person with laboratory confirmation of COVID-19, irrespective of clinical signs and symptoms. Here in the United States, in April last year, the definition of COVID-19 was based on detection of SARS-CoV-2 RNA in a clinical specimen using a molecular technique or assay that is either approved or authorized by the US Food and Drug Administration. So since the pandemic started, up until the end of 2020, there have been now 235 molecular tests that are authorized by the FDA in the United States. 63 antibodies tests and 11 antigen detection tests. Among these are some home collection kits that are specifically authorized to be used together with either one of the antigen tests or the molecular tests. They can be a single analyte for SARS-CoV-2 or in combination with influenza A, influenza B, or RSV. So some of these assays now are still under authorization in the United States. And many in vitro diagnostic companies in the US are working towards getting formal FDA approval of these tests, where they do have to provide more clinical utility and performance characteristics data to the FDA to gain full FDA approval. Of note, there are some also collection kits that are FDA authorized to be used with one of these tests. So they are also in the market. Let's digress a little bit and talk about molecular diagnostic tests. In general, they are nucleic acid amplification tests. Now, the ones that are currently emergency authorized in the US are based mainly on PCR. A small proportion are based on transcription-mediated amplification, such as the Hologic Panther assays, and also some of the rapid tests that are minimal automation, but also very quick, are based on the loop-mediated isothermal amplification, or LAMP for short. So in general, these 250 or so now tests that are molecular based can be done at a point of care with a rapid, short turnaround time with little or no sample extraction. And we expect the results to be available from the time we collect the specimen to getting a result within 1 hour. And some of these perhaps are well known to some of you in the audience, are the Abbott ID Now, the Gene Expert, the Luminex Aries, or the Roche Liat. Now, some of you may have seen this picture of our infamous president in the middle or the peak of the pandemic last year, when he was standing in front of the White House South Lawn, showcasing the Abbott ID Now. So for those of you who may not know, there's actually a small laboratory and clinic in the basement of the White House. And this is what they use to rule out any COVID-19 in some of their symptomatic staff. Now, the other group of tests are laboratory-based. They're of moderate to high complexity. They can be done with or without sample extractions or purifications. So those that can be done directly without preparing or extracting viral RNA are the so-called direct PCR method. And Dr. Wong and Dr. Vail will talk about those with their experience on such a approach to direct PCR testing. Now, these high complexity tests tend to be done in a batch mode. They could be manual, semi-automated, or fully automated. But because they're in batch mode of either 48 or 96 in a run, they take a longer period of time. So can be as short as 3 up to 6 hours to get the results. I've listed some of the examples of these semi-automated or fully automated brand name of the instruments here. So Abbott, Hologic, PerkinElmer, Roche, Thermo Fisher's. Now, these assays differ in the specimen types that are approved or authorized for testing. Some of them are for nasopharyngeal swab or throat swab or oropharyngeal swab, saliva, or some of them, such as the PerkinElmer and Thermo Fisher, are actually FDA authorized to test lower respiratory tract specimens, such as BAL fluid and sputum. One major difference is the sensitivity, the analytical sensitivity, not clinical sensitivity. So analytical sensitivity is the amount of virus that one can detect within the specimens. So in the instance of nasopharyngeal swab, it will be per ml of the viral transport media. So you can see that the point of care tests, even though they're rapid, they are almost a 10-fold less sensitive compared to the laboratory-based nucleic acid amplification tests. Now, even using the same test, there are also differences in sensitivity among the specimen types. And this varies according to the specimen types, but also the time the specimen is collected from first onset of symptoms. So you can see that nasopharyngeal swab or mid-turbinate swab tend to yield higher positivity than anterior nares, the nostrils, or saliva, and then probably the worst is simply the throat swabs. These are some of the meta-analysis showing the sensitivity and specificity. You can see that the MP swab and mid-turbinate swab are almost equal, but then the throat swabs are quite poor in terms of sensitivity. I want to spend a little bit of time talking about antigen tests. You saw in the previous slide that are now 11 antigen tests here in the United States. They're mainly a rapid test based on calorimetric method. So it's a color change when there's antibody bound to the viral antigen present in the respiratory tract specimens. The advantages are that they are rapid. There are no sample processing other than perhaps inactivation of the active virus in the specimens with heat for 30 minutes. They're easy to use, minimal training, and pretty small footprint. So no big, large space needed and pretty low instrument upfront costs. The disadvantage, of course, is price because it's a single use device, most of these. So they're expensive, and typically in the United States, the fancy ones are up to $50 a test. The less expensive, around $20. The throughput is low because you're only testing one sample at a time. And perhaps more importantly, they're not as sensitive as the molecular tests, and because you're not amplifying the target. So if the patient had been symptomatic for quite a few days already, or even if they're before they become symptomatic, the sensitivity is not there. I only have one slide on antibody tests. The Food and Drug Administration in the US have updated its website just last week. And it has been a longstanding policy and recommendation from FDA that serology tests, which is detecting SARS-CoV-2 specific IgG or IgM antibodies, should not be used to definitively diagnose or exclude COVID-19, but it can be used to help identify individuals with recent or prior COVID-19. And just last week, they've also issued, now that quite a few individuals have received either 1 or 2 doses of the Pfizer or Moderna vaccine, is that antibody tests should not be used to assess immunity after COVID-19 vaccination, mainly because of the complexity of the immune response to those who have received the vaccine. So it's quite complicated, whether it's the qualitative test or the semi-quantitative test. The US CDC has similar policies, and they've updated their website, listed down in the footnote here in March. Again, not to be used to establish presence or absence of acute infection, not recommended to assess for immunity following COVID-19 vaccination, or even to assess the need for vaccination in a non-vaccinated individual. I want to share briefly our experience here at our Mayo Clinic laboratories. We are servicing a 1,000-bed hospital as well as 4,000 clients in the United States. So we are national reference laboratories. So March 12 was the first time we launched our in-house laboratory developed tests. We had a potential capacity of 4,000, but because of reagent shortage and supply shortages, we're only able to perform 3,000 tests. So naturally, in the subsequent 6 months, we brought on all these other tests, mainly because each one of them have limitations in supplies and reagents. And so our potential capacity was 38,000 tests a day, but because of limitations of labor, space, and reagents and supplies, we were only able to do up to 30,000 tests a day. I want to share this last slide with the audience, and you can see that this is our epidemic curve since March 2020. We have 3 surges. You may perhaps argue that this is a mini 4th surge, but the biggest surge was around our US Thanksgiving and Christmas period, where the peak cases were almost 300,000 cases per day. And the cumulative death per 100,000 population was at 7. So that's indicated in the yellow. And our test volume during this 15-month pandemic starting in March is represented here. So this is a cumulative bar graph. Blue represents negative tests, and then the yellow or beige color represent the positive cumulative numbers. The bottom graph shows the average positivity rate, and then the line graph represent the weekly positivity rate. So at the peak of our testing, we were doing in November 150,000 tests per week. So far, we've done 4.5, almost 4.6 million tests as of this Sunday. And our peak positivity rate back in November was 16%. But overall, if you look back at the 15 months, our average positivity rate is 7%. So in summary, NAT testing differs in specimen acceptance, workflow complexity, and analytical sensitivity. So the detection rate, positivity rate, depends on the timing of the specimen being collected in relations to its symptom onset, quality of the specimens, transport media, and conditions. And so it's very important for clinicians and laboratorians to have close communications so that they understand what the clinical needs are and what the limitations of the laboratory testings are. I will end there and pass over to Dr. Yan.
Thank you, Dr. Joseph, for the very nice comprehensive overview. We'll go to our next speaker. Our next speaker is Dr. Kristine Alvarado-Dela Cruz, a pediatric and infectious diseases specialist. She has several appointments, one of which is the head Department of Microbiology, Research Institute for Tropical Medicine. She will provide an overview of the COVID-19 diagnostic landscape in the Philippines. Over to you, Dr. Kristine.
Thank you, Ben, for that kind introduction. So without further ado, I'd like to share my slide for my presentation. So a pleasant morning and mabuhay from the Philippines. In this short presentation, I shall endeavor to give an overview of the COVID-19 testing platforms in the Philippine setting. I declare no conflicts of interest and that brands and images that may appear in this lecture does not constitute endorsement of the same. In this briefer, I shall endeavor to discuss the role of the National Reference Laboratories for Infectious Diseases in the Philippines in the context of the country's response to the COVID-19 pandemic. The RITM houses 18 of the infectious disease NRLs, including emerging and re-emerging diseases, and has always been pivotal to the country's response to health security threats. A DOH NRL is a highly specialized facility with technical experts, equipment, and facilities capable of conducting non-routine, highly complex tests in support of the disease prevention and control programs of the Department of Health. Roles of the NRL include confirmatory testing for outbreak investigations, as well as surveillance for disease prevention and control programs of the DOH. The NRL likewise provides support to regulation. The NRL's researches provided scientific data which improved national disease prevention and control program efforts to control and eliminate infectious and tropical diseases, and contributed to regional and global knowledge base of these diseases. Further functions include building expertise for technical leadership, as well as capacity building of medical technologists and laboratory personnel through trainings. By the way, that's me underneath all of that PPE. During the course of the COVID-19 pandemic, the reference laboratories works unceasingly to provide the expertise to support the Philippines' compliance with international health regulations, particularly in the external quality assurance of labs, the conduct of research, kit evaluation, and in the provision of interim guidelines for diagnostics. In 2003, the SARS outbreak was a wake-up call for the country to be ready at any given time for the emergence of a new deadly disease. A national preparedness plan necessitated strong system of detection and identification, surveillance, reporting, and information sharing. Over the years, the NRLs have responded to many outbreaks. As we have all learned, a strong and efficient network of functional laboratories is crucial to provide timely and accurate laboratory services for surveillance and response to public health events and emergencies. Just recently, and prior to the pandemic, several re-emerging infections besieged the country, and it was the surveillance and confirmatory capacity of the NRLs which were crucial to the control and containment of such outbreaks. The laboratory network, technical expertise, and surveillance network system was utilized in its full capacity to be able to provide timely and accurate response in the implementation of effective prevention, containment, and control measures for COVID-19. The emergence of a novel pathogen late 2019 spelled the need for the development of new diagnostics, as well as the proper utility of such. What did the need for new diagnostics for a novel disease mean for the NRL? Success in the nationwide utility of these new diagnostics take the well-concerted efforts of all the stakeholders involved. At the onset, there was only one confirmatory testing laboratory in the country, and an assay was quickly set up and optimized, and at the same time, kit evaluation was conducted as more and more commercial tests were authorized by the Philippine FDA. The surge of specimens overwhelmed the developing institutional capacity for SARS-CoV-2 testing, and RITM had to rapidly reorganize and capacitate itself to respond to the need for confirmatory testing. Aside from this, the NRL also began activating other laboratories by way of assessment, training, and proficiency testing. The subnational laboratories were reactivated all over the country. Nationwide laboratory assessments were conducted, as well as trainings on molecular testing for SARS-CoV-2, trainings on biosafety and biorisk management, fit testing for healthcare workers and first responders, trainings on respiratory specimen collection, and on-site technical assistance and support to regions were conducted, as well as support to logistics for COVID-19 laboratories. As of May 25, 2021, 219 COVID-19 laboratories were activated and with over 30 million samples tested. Antigen tests are part of the COVID-19 testing platforms used in the country, and here you can see the specimen collection activities conducted by the NRLs to validate these tests. Local scientist Dr. Catalina Demetria developed an in-house quantitative ELISA antibody assay. Evidence and science-based interim guidelines were developed and disseminated. To date, the NRLs have validated several in vitro medical diagnostic devices for the detection of SARS-CoV-2. Despite the designation of several tuberculosis labs as gene expert laboratories for COVID-19, and this is basically a sample to result platform opposed to the extractionless format of direct PCR tackled in this forum, and the utility of cartridge-based PCR platforms by some private labs, testing in the Philippines in the majority utilize the conventional plate-based RT-PCR and does not skip the extraction step. To date, there are 29 validated plate-based PCR kits in the country. To give you an idea of the 257 COVID-19 testing labs activated, only 60 are licensed to perform cartridge-based PCR, and that none of these utilize direct PCR. Looking back on where we were before and where we are now, I'd say that there are a lot of lessons to be learned, and there are new things that we are still to discover and hone. But what I know for sure is that in any setting and in the face of adversity, we are truly stronger together. And it is important, such as in forums like this, that we share our knowledge and experiences so that we can move forward with our common goal. Thank you for your kind attention.
Okay, thank you, Dr. Kristine, for that very nice presentation again. So our next speaker is Dr. Eric Vail, a pathologist and director of molecular pathology at Cedars-Sinai Medical Center in the States. He will share his experience with direct PCR. Over to you, Dr. Eric.
Sorry. Hi, thank you. Sorry, let me just share my screen. I believe it's this one. Okay, can you see my slides? Great. So our experience is a little different, a little smaller scale than the previous two talkers, where Dr. Ya was talking about 40,000 a day. We were really being tasked to do 400 or 500 a day. So it's a two log difference. But our main concern was for our inpatient testing, our preoperative screening, and for supplying for the medical network. So just a brief little timeline on how we did this and how quickly everyone, I think, people start to forget about how much of a crunch this was early on in the pandemic, we really made the decision to start to launch our own in-house assay on March 6th of last year, after having a couple talks with Sidney and Rufin at Accelerate Technologies. A week later, we had our first kits, and 8 days later, we had a validated test launched. During that time period, this little room that is about 100 to 150 square feet, or 9 to 14 meters squared. I had to look that up. It was basically our BSL 2+ room. This is where all the pre-processing, all of the live samples were going to be in. And so it was all about finding what equipment would fit inside this room to provide the sort of clean room sort of parameters that were needed. There was significant construction that was done. And at first we just started off with, a hood and a QIAcube doing, 20, 40, 50, 60, 70 samples a day. But by April, we had validated QIAsymphonies and a Roche MagnaPure for a little bit as well, to kind of, as Dr. Yao mentioned, there was significant issues with getting reagents for all of these different platforms. And so trying to figure out how to keep your supply chain going. I don't know if everybody remembered, but in September, the world ran out of tips. There were just no more tips available for anybody to buy. And so it got kind of dicey there for a second. We also had ancillary tests brought online, BD Max, Cepheid, to allow for more rapid turnaround time as well. And then we switched PCR setup, which we had been doing manually up until this point, to an automated pipetter in June. And then we switched to the extractionless PCR that we're using still to this day in November. So standard process. So what we were doing when it was fully automated, after we had gotten people out as much as we could, how was this process set up? Well, we had pre-set up, which means we were printing out labels, creating worksheets, creating batches, manually pipetting into these little tubes. This all took about an hour, hour and a half. Then there was lysis prep and lysis, so pre-lysis before we ever got it onto the machine to start the extraction. The extraction for a full plate was 4 hours. We had 2 QIAsymphonies, so we had 2 running side by side. There was PCR set up for about half an hour, and PCR was about an hour and 15 minutes. So from the time that a batch was started to be created to the time that a result was out, it was about 8.5 hours. And this doesn't account for all the specimen prep before, so the cleaning of the bags, getting everything ready, the transport from collection, all of that is beforehand. And it actually doesn't even account for resulting either. So in that time period, about 2.5 CLS FTE, so clinical laboratory scientists, full-time equivalents, produced about 192 samples, a little bit less because of controls, but just 2 plates. So about 77 per CLS FTE. And it used a ton of equipment and a ton of space and a ton of different places. So when we were thinking that this is just not sustainable, especially as we were going over the Memorial Day surge. So basically a year ago today, we got 2 weeks after that, we got a big surge leading into the 4th of July as well here in Southern California. And we were basically had our techs 6 days a week. So everybody was working overtime. Everybody was working more because we just didn't have enough people to man the instrumentation. So luckily for us, before the big November surge, as Dr. Yao alluded to, we switched over to this extractionless PCR, the Resolute kit. And so the timeline for this is significantly different. Getting the room ready, programming the Hamilton. So we use the Hamilton Vantage. Essentially, we just load the UTM with the swabs in, put it into the Vantage, have that pipette into a 96 well plate, and then or a deep well plate, and then have PCR setup stamped from the 96 well plate into PC, get the whole PCR plate created. This is all done in the Vantage, all done in that same sort of clean or dirty room. And it takes about an hour and 20 minutes. So, 6 hours of time just evaporated like that. PCR is similar, an hour and 5 minutes. And then this means that the batch to result, so pulling samples out is 2 hours and 40 minutes. You're saving about 6 hours just off the bat. And it led to significantly increased production from our CLSs, almost triple the amount of samples could be used with the FTEs. And actually in a crunch, we use 2 people in the room at any given time, just because it is a small room and we prefer to have multiple people in there. But in a crunch, we've actually had 1 person in that room, which actually even doubles that efficiency and that was really helpful when we got this. So these are our testing volumes, our weekly testing volumes, and our positivity rate, really showing across all of our different, what has previously been shown. There was, big positivity, but low testing in the beginning. That positivity went up around Memorial Day, went down, and then towards the US Thanksgiving and Christmas holidays, you had an enormous surge where, we were at 20% positivity. We're doing almost 1,000 samples a day, so doubling our output. But during that time period, because we had switched to the extractionless PCR, our receipt, so this is now receipt to result. So when we got the sample to when the EMR had the result in the system, went down from about 22 to 24 hours to about 12 to 15 hours. And so we were able to significantly, and this is our standard PCR, not even the rapid one, so we were able to significantly cut how quickly we were able to get tests out the door. And I can say that from a lab director standpoint, it decreased the amount of calls that we were getting from the clinicians asking for a test result now, when is it going to be out, things like that. And so this provided us with a real improvement in both our turnaround time, obviously, our clinical performance for the clinicians, and also our staffing utilization. So finally, because this is important too, it doesn't just matter how quickly you can get the results out. What was the performance like? So our CT values were about 2-3-ish cycles higher. So what we had seen previously at 32 cycles was about 34, 35 cycles. And that's completely expected because the extraction concentrates about 4 times. And so if you're doing a log-based 2 system, that's about 2 cycles. But what we found was that during the actual validation is that the analytical sensitivity, so the amount of copies per microliter, didn't go up 4 times. It went about 2 times. And this was because the assay had really excellent reproducibility at the high CT value. So it would just, 38, 39 CTs, it would just always reproduce, especially in the N gene. Often, actually, we'll see things that are 40, 41, 42, 43 CTs, and we'll run them on BD Max, which is by far our most sensitive assay that we have, and they always confirm. I will say, in the beginning days of the pandemic, this would have mattered to me. But as we've watched, as we've seen the sort of natural history of SARS-CoV-2, I have become more and more of a sensitivity skeptic. I think that clinically, it doesn't really matter if you can detect 1 copy per microliter, 0.2 copies per microliter, because symptomatic patients who are at risk to transmit disease do not have one. You could swab outside their nose and you're still going to get positivity. The one caveat that I will say is that because that it is a direct PCR, so you are taking sample directly from UTM and putting it in the PCR setup. About 1 to 2% we've noticed, so 1 or 2 a run, do have a very high background in the PCR, basically unreportable. We used to repeat them once before sending them to other assays. We don't even do that anymore because almost always they don't repeat. Usually it's sample related. There's a big glob of mucus or something in there that needs to be pre-treated or have something else in there. And it just wasn't worth it from our perspective to have that done. So this has been our experience. It's been really beneficial to have that, and it's made my staff significantly happier than they were before we switched over. With that, I'm finished. Thank you.
Okay. Thanks, Dr. Eric Vail, for that very nice overview of direct PCR and the process involved. Our next speaker is Dr. Wong Yu, a medical doctor and founder of Asia Genomics. Asia Genomics Molecular Laboratory is a subsidiary that performs COVID-19 testing. So Dr. Wong will share his experience with direct PCR in the Singapore setting. Over to you, Dr. Wong.
Sorry, just trying to unmute myself to change the screen. Can you see the screen?
Yeah, you can see the screen. Right. Yeah, okay.
Thanks, thank you. So as mentioned by Chair Dr. Benedict Yen, we're a private lab in Singapore. There are about 27 labs in Singapore currently doing PCR testing for SARS-CoV-2 diagnostics. We are one of the private ones. About half of them are what we call belongs to the public or the Ministry of Health, under the Ministry of Health and these are the public hospitals. So our lab itself, we are licensed by Ministry of Health for molecular microbiology, and we also kept accredited for COVID-19 testing in terms of the proficiency testing. Just give a sense, the timing involved, this is a satisfied patient. Some of you will know, he's Eva Ang, is one of our prominent medtech contributors on LinkedIn. He put in his profile on the 3rd of May. He was asked to do a swab because he was at a location that was possibly contact with potential COVID-19 positive patients. And basically, he went to a clinic in Singapore. So if you think about it, Singapore is a primary care system where there are clinics distributed in different parts of Singapore. And he basically went for a swab there. And we provide him a result in about 4 hours, 15 minutes. He liked the timing, he posted it out. So this one, you must remember, there's a few parts to it to come to 4 hours and 15 minutes. One, patient has to get swabbed at the clinic. Someone has to go to the clinic to pick up the swab. It could be 30 minutes from the location. It could be half an hour away from the lab. Once it comes in, as was mentioned by Dr. Vail, you got to do ascension, you got to do the actual run, and depending on where the batch comes in, it may not fit into the previous batch, it may fit in the next batch. You put a PCR run, and then after that, you have to do the results checking before you send the results out. So there's a lot of timing involved, and with a direct PCR, you can actually lower it down to about less than 5 hours between a swab collection, not at your center, at an external place, and giving the result to the patient. So I'm going to go on a little bit about the direct PCR in terms of their advantages. One, it is actually compatible to quite a number of RT-PCR kits used globally. We were actually using a system that is used globally for primary assay, and before we switched to direct PCR. And as what Dr. Vail mentioned, you actually can save about almost 50% time compared to the conventional PCR, because the extraction step for us using our previous primary, it takes about 4 hours from the time we start sample preparation all the way until the end of PCR run. So now what you have is that you actually enable more runs in the same period. It's also very flexible. Basically, now you do not have to use extraction. And extraction kits, as you know, some providers and some vendors actually uses the what we call pre-filled kits. When you have pre-filled kits, you are almost have to complete the entire run, even though you have only one sample to repeat. So that's why I'm talking about flexibility here. You can just run the one sample if it's urgent. So you can actually run that single sample. In terms of cost, it is actually looking at the total cost using extraction plus PCR. The cost for direct PCR is much, much lower. The other point I want to put here is it saves a lot of consumables, because the PCR step and the PCR prep step, there's a lot less pipetting involved. So you don't need so much tips. And also we were using the pre-filled extraction system. In that case, whenever you use only 1 or 2 samples for 16 well pre-filled kits for extraction, you're actually discarding the rest. they are throwing a lot of consumables away. Okay, I just want to give a sense in terms of for the audience here in terms of how you can actually optimize using direct PCR. This is almost similar to what we are doing in the lab. I just want to put a note out there that this is under ideal conditions where you actually have sufficient staff, and you can have staff that will do the pre-accessioning and registration, which takes a lot of time. And of course, you've got to have staff to do all the checking after that. But basically on the wet lab side, the master mix for very trained staff, you can complete it in 15 minutes. That includes the master mix in all the 96 wells. And then after that, a very trained staff, you can actually complete the loading of the entire PCR plate for 94 samples in 30 to 40 minutes. That's our own staff in terms of their training. And in between, you just need one pipetting step. Basically, pipetting, as mentioned by Dr. Vail, a single direct from the sample straight into the PCR plate. So you can actually complete the entire preparation and plate loading within an hour. And on a PCR run site, on a Quant Studio, a Thermo Fisher Quant Studio, it's between 57 to 62 minutes. It's a bit longer sometimes because of the runs. I need to load up the results from the PCR back into the computer system so that you can actually check the results. So if you can actually optimize it, you can actually look at running 3 PCR systems over a 12-hour shift, and you can get about more than 1,000 samples completed. Okay, the PCR runtime, again, just to qualify, this timing is for the Quant Studio. If you're running on a Bio-Rad CFX96, is the recommended time for the direct PCR is about 1 hour, 15 minutes. So depending on the PCR system itself, timing may be a bit different on actual run. Okay, so in terms of disadvantages or considerations, some of you would have used certain RT-PCR kits that comes with interpretive software. From the vendor that we use, the interpretive software is completed for one system. It's a Bio-Rad CFX96. They are currently developing the software for the Quant Studio. One of the reasons is Quant Studio has a lot more noise. For the Quant Studio 5, at least the one we use. So the interpretive software takes a bit of time to develop. It's not completely developed yet. The storage of kits requires -20 degrees, which is actually similar to many kits. What I want to put it here is because, of course, there are some kits you need to store at -80. And then for certain countries, they may not have such a luxury. There are, of course, RT-PCR kits out there that is now lyophilized, where you can just store at room temperature. So I put it as a consideration. And then the actual sample volume, as was pointed out, is only 5 microliters versus up to 400 for certain other RT-PCR kits. So we talk about the accuracy of the test, and that was already brought up by Dr. Vail as well. Interestingly, for this part, for Singapore, because we had low positive rates in the second half of the year, last year, many labs, when they're running the tests, they are not able to get any positive samples to verify. So what our National Public Health Lab, which is under our Ministry of Health, in the fourth quarter of last year, new labs are coming online will have to complete a proficiency blinded test, which means it's a blinded samples that's given to the lab. It could range from negative, positive, and low title. And basically, they ask you to run it and qualify the test back to the public lab. Interestingly, the Resolute test was able to give a much more accurate result for low title versus our original primary. So we're quite confident of this test, as what Dr. Vail has said. So now considerations for starting this. So if you're running different protocols and you have run different systems, there's always the chance of a mix-up when you have different teams working on different systems. So there's a bit of staff training required and internal lab validation. So for us, we are dedicated lab only running the direct PCR, so we completely switch it out. That gives us the chance of mixing up or changing workflows much we decrease the risk of that. So about a little bit of us, we are a fully dedicated lab, only running PCR. We do both single and pooled samples. Pooled samples, we are validated up to 3, we're moving up to 4, so 4 swabs in 1, BSL2+. Our original lab was about 850 square feet in 3 rooms, about over 1,200 square feet. So that allows us to have a clean room, a dirty room, as well as a PCR room. But we have basically converted from a gene testing lab to do COVID-19 testing. As mentioned earlier, we are licensed for clinical lab services as well as CAP accredited, and also proficient for COVID-19 testing because we're running through the proficiency test with the CAP itself, where they send us the blinded samples. Okay, I'll finish here and I'll hand the time back to the chair, Dr. Benedict Yan.
Okay, thanks very much, Dr. Wong. So now we will proceed to the Q&A session. So I just want to state a few things beforehand. The first one is that we apologize that not all the questions that are fielded, either pre-webinar or during this Q&A, where the Q&A that appear in the Q&A box currently. Not all the questions that can be fielded, we will be able to address during the Q&A panel session, but the team will endeavor to respond to every question that has been fielded at a later time point. The second point that I want to make is that some of the questions have already been addressed by the speakers directly in the Q&A box now, as we'll be able to see. I mean, Dr. Joseph Yao has responded to quite a number of the questions, so these will not be fielded. The last thing that I want to say before we go into the Q&A is that I will hand the session back over to Dr. Sidney E in approximately 20 plus minutes or so, for her for the final closing remarks. Right. So with this, we start the Q&A session. So the first questions will go to Dr. Eric Vail and Dr. Wong Man Yu, beginning with Dr. Eric Vail first. Right. So I think there's quite a bit of interest with regards to implementing direct PCR in your labs. So can you comment on the challenges that you encountered during the setup phase and whether this caused any disruptions to your operations? Dr. Vail?
Yeah, so the biggest challenge for us was actually the initial port, and it's not quite related to the PCR kit itself. It was the training the Hamilton to get UTM out of the UTM container and we had 6 different containers of UTM because we were sourcing from all these different places, training it to get the UTM out without picking up the swab. So what was really important was so we had a couple times where we actually had a swab pulled up, spread everywhere. We have to clean the whole machine, and it's a big to do. And so that what I say was our most challenging aspect, and it took a couple weeks of really playing with the machine, getting the vendors in there to get that done right. But once it got started, once the techs got a little more experience with what samples were best to use, because occasionally what they'll do is if they had a sample that's very mucus laden, then what they'll do actually they would take out the swab in the hood beforehand. So they kind of got an awareness, a situational awareness on what samples, how to best treat it. Listen, people are still better than robots. And so that sort of experience, I think, was our biggest challenge. But other than that, the actual PCR itself, the assay itself, it works like butter. It's great.
Thanks, Dr. Vail. Dr. Wong?
Yeah, thank you, Dr. Yan. So our own experience is because we're running our primary with the extraction step. So in order to stop operations, or at least get the staff to say, hey, stay for another hour, we need to validate the test. I think that is one of the challenges because, as you know, staff if they can do less work, they rather do less work, so they can go home earlier. So that was one of the steps. But the training part wasn't too much of a problem because right now, if you're looking at the full conventional PCR where you have the extraction and the PCR, then this is a lot less simpler. The staff actually, once they completed training and they can see that the fact that you can actually run it faster, they get to go home earlier. they wouldn't want to switch back, actually, to be very honest. So that's one. And then to what Dr. Vail said, the same vendor that's actually coming out with our kit that we're using also has the same robotic system. But you're absolutely right. A trained staff can actually do faster in terms of a preparation than a robot can. Interestingly, the robot's a bit slower than trained staff. So, and as you mentioned, a human can actually look at if you're doing poop samples and you get 4 or 5 swabs sticks into one single UTM tube, you will have a problem pipetting the direct sample out. We have yet to get a robot, but we'll see how that goes as we order the system in.
Okay, thank you, Dr. Wong. Maybe I will just add my own question, to sort of for us to take a deeper dive into this, right? In terms of biosafety aspects, because the direct PCR, you have to perform the PCR using the UTM direct or VTM directly in the 96 well plate. Any concerns or lessons to share regarding that? Maybe I go to Dr. Wong first.
So for us, absolutely right. There is always this concern. So I think before every PCR plate comes in from the master mix, we actually wipe down the entire area before we start, so that there's no contamination where the plate sits. And then after that, the trained staff has to do it, of course, under the BSC hood. No direct pipetting, 5 microliters straight into the PCR plate and seal it within the hood. So I think these are some of the considerations that different labs will have to consider in terms of biosafety. But this is what we have been practicing. So far right now, we have done it for the last 2.5 months, 3 months. I think we've been pretty good on that in terms of biosafety.
Okay, thank you. Yeah, go ahead, Dr. Vail.
Yeah, so similar to that, we basically segregated out our pre-PCR or sort of inert area, so that it's just completely in a separate BSL2 plus, sort of its own world, like key card entry, that whole thing. And then when people are in there, they have to be BSL2 plus. That's N95 plus a face shield, back tying gown or PAPR, and double gloves, things like that. And so that sort of consideration, even though it is open inside the Hamilton, so we definitely have a little more exposure than if it was in a hood exactly, but because of keeping everything at that BSL2 plus level, because of keeping everything, so obviously wiping down the sealed PCR plate after it comes out of the room, things like that. We again also have had no issues. I think that any infections in our laboratory were most definitely brought from outside, not within not from the samples themselves.
Okay, thank you both. So now I will address the next question to Doctors Jaime and Kristine. Beginning with Dr. Kristine first. It will be the same question to the both of you. Could you comment on the testing access in the Philippines, the challenges and what other lessons you have learned from that? Dr. Kristine?
So thanks for the question, Ben. So for the RITM in particular, the challenges that we have experienced was heavy on the NRL mandate, I think. So basically, the DOH mandated that all FDA-approved test kits be validated by the RITM. So as you can imagine, it would be quite hard for just one institution to validate all these kits. And it would entail a lot of fresh samples to be collected, some positives and negatives from patients. And this would really, but we have partnered with a lot of institutions for this. So basically that's it. The validation has been a very challenging part of the availability of the kits, of the testing involved in the Philippines.
Yeah.
Okay, thank you, Dr.
If I may add to the, yes, thank you, Benedict. If I may add to the response, on a national scale, I think there were 3 big challenges that we had to face. The first one is for a new disease, is how to upscale the diagnostic capacity or testing capacity. As you would imagine, during the early stages of the pandemic, it was test, test, test. Like everything revolved around testing because people think that before you can do anything, you have to test first. So there was an increased demand and increased pressure on labs to come up with the test results. The second one is how to ensure quality results. You know, it's not just a question of whether you're generating what you're supposed to do, but is it believable and reliable? And then, you know, accreditation comes in the picture. How do you actually network the laboratories? Because it cannot be just done by a single laboratory. And the third one, which I think mattered, especially as the pandemic progressed, is the turnaround time of test results. Because yes, you have the capacitated labs. Yes, you have the quality results. But how long does it take for them to get the results? Again, as you would imagine, people got stuck with the test results in the sense that you cannot do anything until you get the results. I have to admit that during the early stages of the pandemic, the impression was you have to get the results first before you do anything. But of course, you know better that once you suspect yourself to be COVID-19, regardless of what the results will be, you have to be quarantined and to isolate yourself to prevent further spread. But that was not the case. You know, because of so much emphasis on the testing, which I think there was also a communication problem. People were so dependent on tests. Some of them were actually thinking that, you know, until I'm proven to be positive, I just stay put, which is really wrong. So those were really the challenges during the early stages of the pandemic. Thank you.
Thanks, Dr. Jaime. Okay, for the next question, it'll be addressed to Dr. Joseph Yao, Dr. Kristine, and Dr. Jaime again. So please comment on supply chain issues and how you managed, or the laboratory managed to maintain the supply of reagents, equipment, and so on and so forth. Dr. Joseph Yao, please.
Sure. Yeah, it's a common challenge problem faced by all of us in the US. So one way we have dealt with is to approach multiple vendors because each vendor has only so many they can provide. And they also want to keep all their clients happy. So even if they have multi-million-dollar annual business with us, they cannot put all their eggs in one basket for us. I mean, we are one of their favorite clients, but they can only give us so much. So one alternative, which is kind of giving in to the vendor, is that if you buy more machine, we allocate our assays and supplies based on the number of machines that you have. So if you have a need to test more, then you have to buy more of their instruments. So fortunately, and now also very important, is that you have to have your hospital CEO, your administrator, to support you with capital funding to buy. So that's one thing. I think the other thing you can do internally is to have a crisis center group meeting either every day or every week to talk about where can we go and find extra titer well plates, pipette tips, buffers, etc. So it's interesting that our supply chain, our logistics team, and also our lab folks all came together, that we meet daily and talk about and share experiences. And each of them have different contacts. So that's why you saw my slide. We ended up with 7 different assays at the peak of the testing, is so that we can get enough from each of these vendors. No magic, just hard work.
Okay, thank you, Dr. Joseph. Dr. Kristine?
I think Dr. Joseph said it all. It's the same for, I think this is actually a global problem, since it is a pandemic and it's the supply chain really is a problem. Basically, it's maintaining good relationships with a lot of people. So and also it really was challenging for our IDM in particular, because we've been central to distributing most of the supplies for the rest of the subnational laboratories. But it is still a problem for us right now, and it continues to go on. At the beginning of the pandemic, we did also have something like an incident command center that that organized everything, and also had a branch focusing basically on logistics. But it still is a challenge. We basically had to rely on a lot of donations from WHO and a lot of other suppliers for our supply chain. So yeah, that's it. If Dr. Montoya can add a little bit more to that.
Thank you. Dr. Montoya?
Yeah, of course you were knowledgeable on the ground. I'm talking from the national level. My understanding is, there are a lot of players involved when you talk of the supply chain, or having them all to work together is already a big challenge. Add to that, identifying which of the suppliers are reliable. In the Philippines, we have a rather complicated procurement process. If you're going to acquire or secure kits using government funds, it undergoes a complicated procurement process. I say complicated because I'm not even mentioning prolonged procurement process. And remember, this is the standard procedure. And for you to change from a standard process to an emergency or pandemic response, there's also a lag. How do you accelerate the whole process? So, these are enormous challenges. It should take the whole day for me to talk about all of these details, but it was really a big hurdle. And we have to all appreciate the importance of the supply chain to be there when you need them at the soonest time possible.
Thank you, Dr. Jaime. Okay, the next question will be addressed to Dr. Eric Vail and Dr. Wong Man Yu. One of the attendees has commented on the 5 microliter of UTM that is used for Resolute 2, and how that compares in terms of sensitivity to other assays that use hundreds of microliters of transport media. Dr. Vail?
Sure. So first off, remember conventional PCR, like our standard PCR non-kit-based assay, still uses 5 microliters of eluate or something along those lines. So the eluate is 4 times concentrated approximately. So really, you're not really going to, even though you're extracting hundreds to get that smaller eluate, you're not actually running with that 400. Now, the kit-based ones, so BD Max being a great example, you put in like 750 microliters in that, and they're significantly more sensitive analytically. Clinically, it doesn't matter. And actually, it's actually been a kind of double-edged sword in our experience, because we do a ton of asymptomatic pre-surgery testing, and we will get a person that had COVID 6 months ago pop up with a 37, 38, 39 cycle on one thing of the BD Max that it's completely irrelevant. It's not at all clinically relevant. And now all of a sudden that patient's surgery just got, I would love to go back and look and see what the clinical utility, so the benefit or the harm of those low positive results are, especially towards the later end of the pandemic, when tons of, LA County, 40% of people are antibody positive. So everybody's had COVID already by this point. And so it doesn't really, it's not really meaningful to get those super, super low results. And so in our clinical study, we had 100% accuracy with our prior in our validation with our prior PCR. And so I just don't think it's of clinical importance. Obviously, it's scientific, analytical, yes, of course, but clinically not relevant.
Thank you, Dr. Vail. Dr. Wong?
Thanks, Dr. Yen. So as I mentioned earlier, before we were qualified to start the COVID-19 testing, we were actually given blinded samples from our national lab to test, to make sure that we can actually get the results using our previously used primary tests. And we pretty much tested it with Resolute after that. So they went head to head, the results are pretty comparable. In fact, as I mentioned, for a low titer that the primary didn't pick up, Resolute actually picked it up as a low titer. So we were just surprised that it even got a better accuracy compared to the original primary for the particular low titer results.
Okay, thanks, Dr. Wong. Okay, the next question is to Dr. Kristine. is a series of questions. So are direct PCR assays like Resolute 2 available in the Philippines? Is validation outside of the RITM allowed? And is there a recommended route to request for a test with quick turnaround time? Yeah, over to you, Dr. Kristine.
Have to check the Resolute 2.0 in particular. But as to the validation procedures, it's quite standard operating procedure for the laboratory to do validation prior to starting testing. And there are several other universities and research centers who do validation procedures as well. However, it is legislated and mandated by the Department of Health by an administrative order that all FDA-approved kits and procedures be validated by the National Reference Lab. So unfortunately, that has to pass through our validation procedure for that to be used. As of the moment, it's all published in our RITM website. So all the test kits that have been validated by the NRLs are all in that website. So they can check that out there. And I forgot the last question then.
Is there a recommended route for labs that want to have quicker access to a test? Yeah.
Okay, so it's also in the RITM website how to go about the having your test kit approved and your procedure approved. So it's all there. It's all standard.
Okay, thank you, Dr. Kristine. Dr. Jaime, do you have anything to add?
Yeah, well, actually, I wanted to ask a question rather than a comment regarding the Resolute and the direct PCR. If they've actually tried it using saliva, alternative specimens, or pooling of specimens. I raised this issue because especially during the early stages of the pandemic, even up to now, to maximize resources, governments have to look at strategies on how to run samples by batches. And as you probably would imagine, we don't do it in automated fashion. We still do it manually. And if there are a lot of specimens that have to be processed for it to be run. So that's just a question I wanted to raise. I couldn't add any more to the comments of Kristine.
Okay, thank you, Dr. Jaime. Does Dr. Eric or Dr. Wong, do you want to take the question about saliva and direct PCR?
I can take it. As I mentioned earlier, we are validated for both single PCR tests as well as a pooled PCR test. We were validated for 3 swabs in 1 UTM. We're moving up to 4 and 5 in 1 single UTM, so that's pooled. I think the function of how many swabs you can have inside is pretty the size of the UTM before it's all get jammed up. I think the other part about pooled is the current vendor apparently has tested using their own saliva collection kit for Resolute. It's not being used in Singapore yet, but it's apparently validated. You can check with the manufacturer. Thank you.
Dr. Vail?
We for both use cases, it wasn't worth the hassle, to be honest, for us. And so saliva is you'll read the Princeton and the Rutgers of how great saliva is, this and that. And then when you actually use saliva, it's got like a 30% fail rate. And you're like, this is not worth it. It's just not, the headache is just not worth touching. And then pooled testing for us, at least, it was getting brought up right around October as our values were low. And then remember, pooled testing only really makes sense under like 2% like positivity rate. And so when you have super high positivity rate, it's not worth making the pool and deconvoluting the pool and doing all that. And because we're not doing really community screening, we're really just doing our own hospital things. We looked at it and it just wasn't worth it. So we didn't do it.
Okay, thank you, Dr. Vail. Okay, because the webinar is coming to an end soon, this will be the last question that we'll field to all 4 speakers. And this is a question that was posed by one of the attendees. What are the technological trends that are emerging that you see may be relevant to COVID testing? Maybe we begin with Dr. Joseph Young.
Good question. I don't have a crystal ball, but I think our trend, at least in our practice, is to go towards, because at least in the United States, the number of cases have dropped. So the need for large throughput testing will probably decrease. We've actually seen almost a five-fold drop in our tests over the last 2 months. I think it's going to be rapid tests, but unfortunately, the rapid test so far hasn't lived up to its sensitivity. So we are looking for good, rapid, under 1 hour, maybe not single, because you can still have limitations in throughput, maybe 10 or 12 at a time, rapid under 1 hour, pretty good sensitivity, perhaps down to 500 or so copies per ml. I think that's the trend we're looking at. Maybe small plex panel, cartridge base, 1 hour. Somebody mentioned breath tests. So that's actually a very innovative one. The Breathonics in Singapore is going through trial, or maybe has a temporary approval. That's actually very innovative, but I don't have any data about its sensitivity.
Thank you, Dr. Yao. Dr. Kristine?
Okay, so my take on this is that right now I think with the trend in diagnostics, the focus should be more on other things, because it's been 1.5 years in this pandemic. So we're focusing not mainly on detecting cases, but rather on vaccine surveillance, things like that, and whole genome sequencing, and basically that's the focus for now. And I think the trend for the diagnostics should follow that need. So that's my little take on that matter.
Okay, thanks, Dr. Kristine. Dr. Vail?
Sorry. So I definitely want to echo those points. I think that we have to remember that we're not really preparing for COVID testing anymore. We're preparing and looking at what the testing for the next pandemic is gonna be like, and what are the lessons that we learned from this one. And I think that this is kind of be sort of US centric, but looking at how badly the FDA bungled the initial regulatory landscape of the testing, and how they made it as difficult as possible to get a test launched in the early days. I think we're going to see a lot less of that than the next time around, and a real loosening of the ability to get innovative and high quality testing out. Hopefully.
Okay, thank you. Thanks, Dr. Vail. Okay, last one, Dr. Wong.
So I just want to echo a little bit about what Dr. Yao said about the breathalyzer test from Singapore. So I have a bit more information since I'm based here. So there are this is actually quite a really interesting area where it's actually looking at the chemicals from the breath that is as a surrogate marker to an actual diagnostic, where you're actually detecting the actual virus. So you're looking at breath markers, what you breathe out, right? So we have found out that whether you have cancer or you have a smoker, the chemical composition of the breath is actually pretty different. So this whole area of breath omics or something is like genomics, right? It's coming to an interesting start in the whole industry. So I think it's an interesting place to watch. You can use it as mass screening, certainly. Not diagnostic. It has to be a diagnostic test following that. But I think this will be interesting, not just for the COVID, but also for other pandemics that may come about. Yeah.
Okay, thanks, Dr. Wong. Sorry, I forgot to, maybe the last question should also be directed to Dr. Jaime, who will then end the Q&A session. Dr. Jaime, please, your thoughts on technology trends.
Yeah, well, I couldn't say more to what was already mentioned, but from a developing perspective, whether you're going to look at the surveillance for surveillance purposes or for actual patient care, I think the trends are evolving towards something that is simple, rapid, reliable results with a short turnaround time, with not too much technology or manpower dependence, that can be interpreted easily and can be correlated by clinicians to what is actually happening in the patient. And I should say, with the variants coming up, if I can throw in the variants question, because as of course we know that there's a lot of interest now in variants, and how does it impact on diagnostics? Do you actually have to do genome sequencing for all of the patients that you see? So maybe that's one trend that we're looking forward to. I mean, because of this interest and occurrence of so many variants, for example, which I'm saying is not all clinically important, but some of them will be, as they call it, variants of concern or variants of interest. Thank you.
Thanks, Dr. Jaime. Okay, and with this, we end the Q&A session. Thank you to all our speakers and Dr. Jaime for a very engaging and lively conversation. I hand it over back to Dr. Sidney E now.
Yeah, hi, thanks. Thank you, Ben and the panelists for this very vibrant discussions this morning. So just to put in context, although today's topic is on direct PCR as an alternative platform, but the webinar is really about alternative testing tools in general. Direct PCR really is just one of the tools. And I guess it's not surprising because there isn't really such a thing as a one perfect test for all use cases. It's important to understand the context of our use case and decide on the best tests to deploy for our purposes. I mean, for example, do we want to have fast turnaround time? Do we want to have a high throughput? And as a result of that, do we need to compromise on a little bit on the performance, or whether it makes sense to go to CT beyond a certain number, how relevant it is clinically. So I think those are all different use cases that would require us to think about in terms of the type of tests that we are looking at. This is, of course, especially true in this pandemic where the clinical manifestation of the virus is evolving. Dr. Montoya mentioned about all these VOCs that are evolving, continue to come. We really need to stay ahead and get our tools ready. And I think this is really the gist of this preparedness webinar series. I noted that there are many more questions posted by the participants. It's just really unfortunate that we didn't have time to go through and discuss all the questions. There are just too many questions to go through in such a short webinar, but don't worry. We will address all the questions on the ASEAN Dx website, and we'll post the website after this webinar, at the end of this webinar. We hope to continue the discussions there. For now, to help guide us on what topics matter the most to you where diagnostics testing is concerned, we're going to run a short poll. I'll flash the questions on screen, and so we'll leave them on for a minute. So it would be helpful if you guys can go through that in the next minute or so. So I think we are just about done. And so thank you so much for responding to the poll. The result of the poll will be made available on the ASEAN Dx website. It will certainly help us guide what sort of topics we want to talk about in the next preparedness webinar series. So as this is really an ASEAN Dx platform, we also would like to include a lot more representations from other ASEAN countries, ASEAN member states. Very grateful that for this first of the webinar series, we're able to have Philippines with us. We would love to be able to invite more of the other ASEAN member states to be with us. A big thank you again to the panelists, and thank you to all of you for staying with us through these discussions. We hope to see you again at our next preparedness webinar. Thank you and stay safe.
Yeah, thank you very much to everyone. And congratulations, wonderful meeting, Sidney.
Thank you.
Thank you. Thank you, Jaime. And
Thank you.
to all the panelists.
Thank you.
To all the panelists, thank you very much.
Thank you so much.
Thank you. Have a nice